Most welcome my friend in our channel. We are going to understand basics of the genetic engineering. Most of my students, either the MSc at graduation level, a BSc level, C undergrad, like senior secondary schools, they need to understand what the basics concept of genetic engineering. We will start from its basics, from who can discover, who was the father of genetic engineering, and basic principle behind them. First we need to understand clones. Clones means that identical copies. How to develop these clones we need to understand in later sections. And genetic engineering, also called the recombinant DNA technology, rDNA technology. So we need to one by one understand steps. This is the new discipline of science which is used to manipulate, that can need to change according to we require the genes in the next generations in hereditary materials. Either the gene, the functional part of the DNA is called genes. If we want to manipulate, we want to insert something, new things, new character, so we can change that particular specific genes in this technological applications. It includes the set of molecular techniques and formation of the recombinant DNA or DNA. Hence, is this technique also called the recombinant DNA technology. Already I told you, this is made possible by forming clones. How can you make? If you want to make these things, you need to make clones, and these clones are giving what we need to achieve the gene clonings. This technique was first developed by the Paul Berg in 1971, who made the first attempt and manipulate the DNA, and he construct the first RNA, first rDNA molecules by using the virus, simian virus, where they can use this virus, this genome, into bacterium cells. That's why he is also called as the father of genetic engineering. So we need to understand Paul Berg is the basics, regulate and teach how you can change a genes according to we need require. The process of gene cloning is a stepwise process. First step of process, the cloning, means produce the identical copy by manipulating genes inside the genetic structures, and then we need to isolate the required genes, which genes we need to go for the application changes, we need to isolate, then synthesize the recombinant DNA molecules by techniques, recombinant technology. Now this rDNA, recombinant DNA, introduced into the host cells. This is another steps, and then these host cells go for the replication with their DNA and this rDNA molecules. So we understand these basic steps are present in the gene cloning. First is the production of the clones, identical copy, number of copies, like PCR machines, we produce large number of the DNA molecules. So just like that, we also need to require the specific DNA molecule which is required for the clonings. Then we can forms within these clones, they can make rDNA, a recombinant DNA. Now this recombinant DNA introduced in the host cells, from where this recombinant DNA with the help of host machinery DNA replicate. So the isolation requirement of the gene, this is the most important steps. Isolation of required genes, desired genes bolo, we are going to understand gene of interest bolo, the donor DNA, foreign DNA, passenger DNA, we need to call many names of these things. The techniques would need for the isolation, generally we are using artificial selective method. In this method, genes required synthesized inside the lab, then isolate, either artificial synthesis, reverse transcriptase method. You know reverse transcriptase method, that can be synthesized gene from the mRNA molecules. Another method is shotgun method. Shotgun method is a very recent method. It includes the random collection of the comparative large number of DNA fragment of the genes and then go for the required gene isolations. Now in this whole process, one enzymes that play major role is the restriction enzymes. What the role of restriction enzymes? Why it is important? Because the restriction has the only work on double stranded DNA molecules. They cut the strand of double stranded DNA molecule and they produce the palindromic sequence. You know palindromic sequence, you can Google on, then you find the basic things of palindromic. This is the character of restriction enzymes. And molecular side, they specifical cut, they can cut like a scissors, microblade is also called, chemical knife is also it is calling. Sticky end generate and cut. Because of these restriction enzymes have these type of the characters, so they can produce sticky ends. So if we are going to recombinant formation, so we need such type of the sticky ends. These palindromic sequence, specific sites what we require for the cloning. Some exceptional, we have also used transferase enzymes that cut the blind end, not the sticky ends, this type of restriction enzyme. In order to recognize cut as isolation of double stranded restriction enzyme, endonuclease enzymes use molecular seizures that can be understood. This is called, this enzyme is a site specific, already I told you, very specific, and so many other things in this restriction enzyme. That's why very important in the gene cloning methods. Now after the restriction digestions, we need to go for the RNA DNA, recombinant DNA molecule synthesis. So if there is a clones are there forms, so we need to cut the specific, there is a sticky information and required specific type of the genes are present, then we go for the bind with them and produce the specific recombinant DNA molecules, and that recombinant DNA molecules is required for the our genetic engineering, genetic clonings. Cloning circular DNA or plasmid, generally we for cloning clones, that need for the this process is the plasmid, either from the bacterial plasmid or any other plasmid that have the capability, they can go for the replication with these foreign DNA molecules. So here you can see this is the rDNA molecules and this is recombinant donor DNA molecule, specific DNA molecule, vector molecules. This vector is called the plasmid vector. Now the enzymes required for the binding of these is called DNA ligase enzymes. This is joins the required genes and foreign DNA, this vector DNA, and make recombinant DNA. Some restriction enzymes cut the blind end, they can made sticky by terminal transferase. If you can find any type of restriction enzymes that made a blind end, not the sticky end, so we use another enzyme. This enzyme is called terminal transferase. They can cut like a sticky end. So these sticky end bind with vector DNA molecule, that is closed circular DNA, plasmid DNA, where the sticky ends are present, they can bind with them and produce rDNA molecules. So now you are ready, these rDNA molecules are ready to introduce in the host cells. The host cells are bacteria such as E. coli in the place in the mediums. If there is a mediums where the growth of this bacteria is common, dilute solution of calcium chloride is in order to produce bacterial cell walls. So this type of the mediums require for the other type of the growth of bacteria. So you know that calcium chloride does also make bacterial cell wall permeable, so the foreign DNA molecule from it go inside the bacterial cells, and where they combine with their plasmid and form the recombinant DNA molecules. And DNA of bacteria now ready for this cloning process, because this is the this is for here we go for the in this steps we are going to understand if diagrammatically replication of the DNA molecules with the host molecules. Finally the cells are ready, just like that. This is the vector DNA and this is the donor DNA, and this is the restriction enzymes. We cut this DNA molecule, sticky information is here, and this DNA required also cut by this, both cut by the similar type of restriction enzymes, so they produce similar compatible stickies, so they can form, when they are going to join with them with the help of ligase enzymes, they form the DNA rDNA molecules. This rDNA molecules introduced in the E. coli go for the transformation process, allow to replicate, and they produce the clones. These clones are identical type of things. So this is the whole process of the genetic engineering, very simple and very understanding enables how you can understand genetic engineering. Genetic engineering is a basics. You can make a DNA into number of other DNA with specific characters, identical characters are required number of the characters, so you can produce next generation with that specific characters. So genetic engineering is more frequent research in the upcoming generations, so we need to produce number of proteins, number of the organs, number of things required for the human welfare. Hope you like my video. If you really like, please subscribe my channel. Thank you so much for watching.